Okay, so this is the video for lecture number 18, and in this lecture we're going to get into statistical tests for when your dependent variable is a numerical variable. This is going to be the last week of new material in this course, and then the week leading up to the final exam is just a chance for you to study and review and come to help sessions. Okay, so I want to start with this situation here. So imagine I ask 200 students in this class to guess a series of 5 coin flips in a row. So I'm going to flip a coin 5 times. Before I flip it, you're going to write down your guesses, and if you guess right, you'll get some big prize. And so I go ahead and I flip my coin 5 times, and we're going to check each student's answers against my coin flips and see who got it correctly. And so a question for you is it very likely that you would guess all 5 of the coin flips correctly? What do you think the probability is that you'd be able to do that? Well, it's pretty rare. The probability of any one student guessing perfectly for all 5 of the flips in a row is a half to the power of 5. So the probability of guessing any one coin flip correctly is 50% or half. And then to be able to guess those 5 independent flips correctly, you have to multiply each of those halves by itself. So you end up getting a value of a 3.1% chance. So it's possible, but pretty rare, right? Now, given that it's rare for you to guess all 5 correct, do you think it's very likely that at least one student in the class would guess all 5 correct? Well, the surprising thing is it's practically guaranteed that at least one student would. So for any one student, again, the probability of guessing less than 5 correct is 1 minus 0.031, or 100% minus 3.1%. So there's about a 97% chance that you don't get all 5 correct, right? So that's the probability of you guessing something less than 5 correct. The probability that no student in the class guesses all 5 flips correct is the same as the probability that all of the students in the class get 4 or fewer correct. And so we can calculate that by taking 0.97 multiplied to the power of 200. So sorry, I should have said multiplied, but multiplied by itself 200 times. Okay, so that number is the probability that no student in the class guesses all 5 flips correct. So then all we have to do is take 1 minus that number, and that gives us the probability that at least one student in the class gets all 5 correct. And it turns out that 1 minus 0.97 to the power of 200 is about a 99.77% chance that at least one student gets all 5 correct. So think about this for a minute in terms of significant p-values and false positives. It's relatively rare to get a significant p-value when the null hypothesis is true, right? That's determined by our alpha level. If we set our alpha level at 0.05, that means that when the null hypothesis is true, we'll get a false positive 5% of the time. But what if we perform the same study over and over again? The more times we perform the sample and the analysis, the greater the chance that at least one of those times will get a statistically significant result. And in fact, if we do it 100 times, we're practically guaranteed to get a statistically significant result, probably a few times. So now imagine you're a company that is like marketing a new cold medication or something like that. Well, you could repeat your study lots and lots of times until you get the result you want and just publish that result. So do you see the problem here? There's a problem of doing lots and lots of tests and not reporting all of them. Okay, so when you perform many tests, the probability of making at least one type 1 error rapidly increases. And we can plot out what that probability looks like because it's just calculated based on the alpha level we choose. So if we choose a standard alpha level of 0.05, this is what that curve looks like. So the more times you do a test, the greater the probability that you'll get at least one false positive. And by the time you're up to like 50, 60, 70, 80 tests, it's almost guaranteed that you'll get at least one false positive. And so this is the issue that came up some years ago with the cold effects medication. It was found that the company was selectively reporting and only reporting the tests that showed the conclusion that they wanted. That's a problem. Okay, so the more tests we run, the more likely it becomes that we'll get at least one positive result that is false. And this is sometimes called data dredging, p-hacking, or fishing for a positive result. And the point is that it's misleading to do lots and lots of tests, but only report the significant ones. And why? Because it misrepresents the type 1 error. If you only tell the readers about the tests that were significant, you're misrepresenting the true kind of distribution of results. So transparency is really important. Now, there are situations, especially today in modern research, where we really do need to do a lot of tests. And here are a couple of examples. The first one I have is genome-wide association studies. These are more and more common as we have genome, like the human genome for decades now, and many, many other genomes in other organisms. As an example, we know that Alzheimer's is a disease that has heritability. So it tends to run in families. And so that indicates that there's potentially genetic variation that contributes to the risk of Alzheimer's. So researchers want to know, what are the genes that might be causing it? Because if we can identify those genes, we might be able to find a cure. Well, the human genome has thousands and thousands of genes. So we can sequence the genomes of people who have Alzheimer's and people who don't. And then we can run thousands of statistical tests looking for associations between each of the genes and Alzheimer's. So in each test, the genotype of the person is the independent variable, and whether or not they have Alzheimer's is the dependent variable. Well, when we run thousands of tests like that, we're virtually guaranteed to get a whole bunch of false positives. We'll probably get some true positives as well, but we'll get a bunch of false positives. And that's just a feature of statistical testing. It's just a feature of random variation in a sample. And it's a feature of the null hypothesis testing paradigm. But we need to be able to answer questions like this. So what do we do when we need to do these thousands of tests? Well, there are modern techniques to account for this. One technique that is based on the null hypothesis paradigm is called false discovery rate. We're not going to get into the details of how it works, but basically what it does is it adjusts the threshold for statistical significance depending on how many tests are performed and depending on the distribution of p-values obtained. And it's the best choice for studies that need to do many, many tests, like hundreds or thousands of statistical tests looking at the same question. It's appropriate to use a false discovery rate adjustment. So it keeps the overall rate of false positives within a given study to about 5% on average. So it doesn't mean that you get no false positives, but it just gives you a more realistic idea of which of your results are likely to be false positives and which are more likely to be true positives. Okay, so we're going to get into a couple of different kinds of tests for when your dependent variable is numerical now. And let's start with the question of what if we want to compare more than two groups? And this is related to this multiple testing problem. And I'm going to give you an example using an experiment that I did with the students in 2019 to test whether different methods of note-taking would affect the students' abilities to remember a bunch of words. And you've seen this experiment before in our lectures. So the dependent variable in this experiment was the number of words that a student correctly remembered. And the independent variable was the method that I told them to use to try to memorize the words. And I'm not so worried about the details of the different methods that they used, but let's say we had 4 different study methods, method 1, method 2, method 3, method 4, and here you're seeing box plots showing you the students' memory scores. So higher scores mean that they remembered more of the words, and you're seeing the distribution of those scores for students in the 4 different groups. So how could we analyze this situation? Here's another example of the graph shown as a dot plot instead of a box plot, so you can see all the data points. Well, you might say, well, we could do a bunch of 2-sample t-tests, right? So in each 2-sample t-test, we'd be able to compare two of the methods to each other, because the two-sample t-test, we can only compare two groups. So a question for you, how many two-sample t-tests would it take for us to do all possible comparisons of these four groups? We'll take up the answer on Zoom. It would be quite a few. And a more important question, why is that a bad idea? Why would it be not a great idea to run all of those two-sample t-tests to try to answer the question of this experiment? Are these different study methods do they differ in terms of how well people are able to memorize? Okay, well, we don't have to do that. We don't have to do all those two-sample t-tests. There are other kinds of tests we can use, and the one we're going to learn here is called ANOVA, which stands for Analysis of Variance. It's a single test that simultaneously compares the means of two or more groups to see if the means are different. So it's kind of like expanding our two-sample t-test to have potentially more than two groups. It needs a numerical response variable, and the test assumes that the numerical response variable is normally distributed. The data are, as always, assumed to come from a truly random sample. And here are some examples of questions that can be addressed with ANOVAs. Is there a difference in blood pressure between patients who got drug A, drug B, or a placebo? Is the average sugar content different when comparing 4 different batches of beer? So I hope you can see we've got this categorical variable, which batch the beer came from, and we've got 4 groups. So we can't use our two-sample t-test, but this is appropriate for an ANOVA. Another example would be, is the average daily snowfall different in Winnipeg, Ottawa, and St. John's, comparing these 3 different cities and our numerical measure of snowfall per day? Okay, so a few more assumptions or requirements of the ANOVA. Each group, again, is assumed to be a random sample. Each group should be normally distributed, the response variable in each group. And each group should have about the same variance or spread. So ANOVAs differ from t-tests because ANOVAs can have 3 or more groups, but ANOVAs can also be done if there are only 2 groups. And in the case of 2 groups, the ANOVA would give us the same conclusion and usually the exact same p-value as the two-sample t-test. The null hypothesis for the ANOVA is that all of the group means are equal. For example, for our memory experiment, the null hypothesis is that the mean of method 1 is equal to the mean of method 2 is equal to the mean of method 3 is equal to the mean of method 4. Okay, so let's get into what an ANOVA does. If the null hypothesis is true, the differences or the variance between the groups should be less than or equal to the differences or variance within the groups. And so looking at these kind of cartoon histograms, the variance within the groups is like the spread of each group histogram, and the variance between the groups is the spread or the distance between the group means. And here we have a situation where the variance between the groups, you can see, is much bigger than the variance within the groups. Right? So in the top situation, that's what we expect to see if our null hypothesis is true, whereas in the bottom situation, that's what we'd see if our null hypothesis was false. So the ANOVA works by calculating a test statistic. The software does this for you, called F. It's the F ratio. It's the ratio of the between group variance to the within group variance. And so the top situation is one where F is less than 1, because as you can see, the between group variance is relatively small and smaller than the within group variance. Whereas on the bottom, this is a situation where F is larger than 1. And the p-value for ANOVA compares the calculated value of F based on your data with the expected distribution of F under the null hypothesis. Okay, so we covered that. And so what happens if we reject the null hypothesis? What do we do with an ANOVA if we get p of less than 0.05? Well, we reject the null, of course. And what that means is that we've negated the kind of line where I said mean group 1 equals mean group 2 equals mean group 3 equals mean group 4. It tells us that at least one of the groups differs significantly from the others. If we reject our null hypothesis with our ANOVA, at least one of the groups is different. But we can't tell which one or how many are different from each other. Like it could be that all 4 of our groups differ from each other, or it could be that just group 2 differs from group 1, 3, and 4. We can't tell just from running the ANOVA initially. We need to do further testing to identify the kind of pairwise comparisons. So when people use this kind of test, obtaining a statistically significant ANOVA in the first case gives us justification for further testing to compare specific groups. We're not going to get into that in this course, but when you do further tests of comparing pairwise comparisons of these groups, that's often called post hoc tests. If you're curious, you can ask me about them, but we won't cover that in the class. Okay, so there's another important statistic that you can get out of the results of an ANOVA, and that is called R squared. R squared is also called the coefficient of determination. That doesn't help me remember it. But what it tells you is R squared tells you the proportion of variance in the response variable that's explained by the predictors. And so for our ANOVA, it's telling us how much of the variance in students' memory scores is explained by which study method they used. So R squared can range from 0 to 1. It can be expressed as a percent by multiplying that value by 100. So you could say, okay, R squared was 50% or 0.5. And R squared of 0.5 means half of the variation in the response variable is explained by the predictors. Okay, so I already said that part. So when we report on our ANOVA, there are several things that we should report to fully describe the result, including its effect size. We should talk about or present in a table the means of the groups and their spread or their variances. We should report the F statistic, the p-value, the sample size, the type of test, and the R squared value if the result is significant. It doesn't really make sense, I don't think, to report R squared if you have a non-significant result. So here's an example of an ANOVA, reporting the results of our experiment that I did with students in the class. I've got a graph where I've represented the means and their 95% confidence intervals using whiskers. And so I might say, in our experiment, we did not detect a statistically significant difference between the 4 different methods of studying in average memory performance. Refer the reader to Figure 1, tell them the type of test, ANOVA, report the F statistic, the p-value, and the sample size. So I didn't report the R squared, although in this case I could have calculated it. It was 1%, but I didn't bother reporting it because we had a non-significant result. So our results were consistent with our null hypothesis here. Okay, so let's take a look at this example that we saw in a previous lecture of analyzing differences between male genotypes and their kind of pair bonding score in this particular study. So they had a very statistically significant result, 0.0004, but the R squared in this case was only 0.02. So even though the study found a highly significant difference between these 3 genotypes, the genotype explains almost none of the variation in pair bonding behavior. So I just told you the answer to this, but practice writing out, how would you write out the results to the study, including reporting the R squared? So can you summarize the results of this test in a sentence or 2? And we'll take a look at that on Zoom. This is the kind of thing that you'll definitely be asked to do on the final exam. Okay, so how do we analyze the association between 2 numerical variables? We're going to get into the last type of test that you're going to learn in this course. Well, a correlation is a type of statistical analysis used to quantify the relationship between 2 numerical variables. And we've seen this image before where we've got 3 scatter plots showing you 3 extreme scenarios for this kind of a relationship. You could have the far left example, which is no correlation. You could have a negative correlation where the value of y tends to decrease as x increases. Or you could have a positive correlation where the value of y tends to increase as x increases. The correlation coefficient R is a statistic that measures the strength of the linear association between 2 numerical variables. The correlation coefficient tells us how well the data points line up on a straight line, and it tells us whether they're correlated in a negative or positive way. So the correlation coefficient R can range from -1 to +1. It has no units, and it's beautiful because it can be compared across many different studies and topics. For example, it's great for being used in meta-analyses. If there's been many studies looking at the relationship between two numerical variables, like say body weight and metabolic rate, you can extract the correlation coefficient from all of those different studies, and it doesn't matter what those studies, what units they measured their variables in, the correlation coefficient is comparable across all those studies. And remember, an R value of 0 implies that there is no linear association between the two variables. Okay, so the maximum possible absolute value of R would occur if all of the data points fall on a perfectly straight line. We really never see examples like that in biology, although sometimes it comes pretty close. So that would mean that we had a perfectly linear relationship. As shown here, we've got a perfectly linear negative relationship and a perfectly linear positive relationship. Now I want you to notice here, though, that the linearity of this relationship does not depend on the units of x and y. We could change the units. We could add 10 to every x value. We could divide every y value by 3.3. We could do any transformation we want to x and y, any linear transformation, I should say, and the relationship between x and y would have the same R. Okay, so the linear relationship is independent of the kind of metric units, for example. Okay, so now we're getting into this other type of test, a correlation test. So this is for looking at whether there's an association between these two numerical variables. We've got some assumptions. The variables are assumed to be normally distributed. The data, as always, are assumed to come from a random sample. And here are some examples of questions. Is there a relationship between brain size and body size across humans? Do animals with larger eyes have longer legs? Is the average daily snowfall associated with the average daily air temperature? So a few more assumptions and requirements. The correlation test assumes that there are no major outliers. Crucially, it assumes that the variance or spread in the y variable is relatively equal across values of x. So it would not be appropriate to fit a correlation or to run a correlation test if you have these kind of funnel-shaped situations where the variance in y changes with values of x. It's also assumed that the relationship is linear, that it makes sense to look at a straight line for this relationship. And so that's one of the reasons, well, these two examples are reasons why it's so important to visualize the data before running the statistical test, because we wouldn't want to run a correlation test on, for example, the little image on the lower left, where clearly there's a curvilinear relationship between y and x, and it just wouldn't make sense to analyze the straight line relationship between those two variables. So that's why it's important to always look at the data first. We're not going to learn tests for these curvilinear relationships in this course, but they exist. There's lots of other methods out there to use for those situations. And the null hypothesis for the correlation test is that R equals 0. Okay, so here are some examples of scatter plots, 3 different situations of y and x's relationship. And the question is, should we run a correlation test in these examples? Well, no, not in any of these cases, because all 3 of these cases represent cases where the spread or the variance in y changes with values of x. So they're violating this assumption of having consistent spread across x values. This change in variance across values of x is referred to as heteroskedasticity. So heteroskedasticity just means like the variance is variable. Okay, it can be diagnosed by seeing this kind of funnel shape or a bow tie shape in the scatter plot. And if you see data like this, it might mean that you need to transform your data or use another type of analysis, which we're not going to cover. Okay, so R squared can be calculated for correlations too. So remember, R squared tells you the proportion of variance in the response variable that is explained by the predictors. And to calculate R squared for a correlation, you simply multiply the correlation coefficient R by itself. So it's just R squared, and that gives you R squared. R squared ranges from 0 to 1, as we saw before, and it can be expressed as a percent. And just like R, it can be readily compared across studies. So let's quickly look at an example of a correlation analysis using data that I collected from students in this course in a previous year. And I asked students to report how tall they were, and I also asked them to report the height of their best friend and the height of their siblings. So one question we can ask is whether a student's height is correlated with the height of their siblings. So does height tend to run in families? Well, we can address that with a correlation analysis. We can also look at this question of whether people's heights tend to be correlated with the heights of their friends. So do taller people have taller friends on average? Do shorter people have shorter friends on average? Again, we can look at this with a correlation analysis. So you can take a look at the data we collected from the students. You can even download it and plot it yourself. If you go to the link on the bottom left there, you get the raw data, and then you get the version that I tidied up on the right, because students entered their heights in all kinds of different formats. So it needed some tidying. Okay, so here's a graph of student heights on the x-axis and their siblings' height, if they had a sibling, on the y-axis. And here's a graph of student heights on the x-axis and their friends' heights on the y-axis. They had to kind of guess what their siblings' heights and friends' heights are. I hope you notice right away there's something funny going on with this data point here. And some students entered some commentary in the data. And so this student's best friend was their very cute dog. So I think we want to exclude that data point. So we need to do some data hygiene here. And so this is what the data looks like after we exclude that one. So we want human friends. Okay, and so we can run the correlation analysis on the sibling student heights, and we get an R of 0.24. So that's a positive correlation. It's fairly weak. In fact, it's not statistically significant. We can get a 95% confidence interval on R, and we can get the p-value and our sample size below. And so we might conclude we did not detect a statistically significant correlation between the height of a person and his or her siblings in our sample. And then we can report all those kind of quantitative details. On the right, we run the same analysis. This time we got an R value of 0.41. It had a 95% confidence interval of 0.14 to 0.62, and the p-value for our test was 0.004 with a sample size of 48. So here we actually reject the null hypothesis. The p-value is less than alpha, and so we can say there was a statistically significant positive correlation between the height of a person and his or her best friend in our sample, and then we can report the details in the brackets there. Okay, so here I'm plotting those two correlation coefficients as a point with their 95% confidence intervals to kind of show you what we're concluding here. So in both cases, the R value was positive, but in one case, the 95% confidence intervals overlapped with our null hypothesis that R would be 0, and in the other case, the confidence intervals excluded that null value. So I just want to highlight how useful this is. 95% confidence intervals, we practiced calculating them on the mean of a numerical sample, but confidence intervals can be used for lots of other statistics, such as R. And so that's what we're showing here. And just remind yourself, what do these confidence intervals tell us again? It's a measure of uncertainty around our estimate of R. And it means that if our sampling procedure was repeated lots and lots of times, these whiskers should capture the true population value about 95% of the time. So we don't know what the true population value is, but the point is that this shows kind of a plausible range for the true population value. Okay, so here's some data from a much bigger study looking at some similar questions. So this was a study done, I think, in Sweden, looking at correlations among siblings for a bunch of different traits. So feel free to critique this graph. It's got some issues for sure. So we've got like 5 different colored data points here representing R values. We've got correlation coefficients R on the y-axis, and along the x values, or along the x-axis, we have different types of siblings. So on the far left, monozygotic twins, that means identical twins. I'm not sure what the difference is between monozygotic and dizygotic twins. Oh, I guess dizygotic twins just means twins who shared a womb but aren't identical. They don't share a genome. And then the third category here is our full siblings. So that should be the most comparable to the study we did of students in 1105, which would mostly probably not be twins. And then we've got other categories of like siblings that were raised apart, half siblings raised apart, and then adopted siblings, which are children who would have grown up together but would have different parents. Okay, so this is the category we want to focus in on. And here's our result. That's the correlation coefficient we got with its 95% confidence interval. So you can see that our uncertainties were very big on our estimate. But interestingly, the correlation coefficient that they obtained in this study for height was a little bit outside of the range of our 95% confidence intervals. Now, one of the things about their study was it focused on same gender siblings only. And so that might explain why our sibling correlation was a little bit lower than what's been reported in previous studies, because they limited their analysis to just siblings that had the same gender. Okay, so I want to just show you a couple of examples for why we need to report all of the details when we're presenting the results of these analyses. So I'm going to show you a couple of scenarios here. In the first one, let's say we read that the rate of ice cream sales per day is positively correlated with the rate of road rage incidents per day with a correlation coefficient of 0.34. Well, without any more information, it could mean all kinds of things. It could mean something like the graph on the left. It could mean something like the graph on the right. Only one of these two graphs is statistically significant. Okay, here's another example. The rate of ice cream sales per day is positively correlated with the rate of road rage incidents with a p-value of 0.02. Well, again, that could mean any number of things. That could mean something like the graph on the left here, or it could mean something like the graph on the right. Both of these are consistent with what's written over on the left here. And so in this middle example, it's a much stronger correlation, the one on the left, than the one on the right, even though the p-value is the same. And so that's why it's so important to report all of the sources of information that you need to convey your results. So here's a third example. The rate of ice cream sales per day is positively correlated with the rate of road rage incidents. Report the type of test, report the correlation coefficient, report its 95% confidence interval, the p-value, and the sample size. And so that would uniquely identify a given strength of correlation and a conclusion about the null hypothesis. And so here's the kind of best case scenario where all the information is reported. And it's useful to show the data as well so that readers can make sure and kind of investigate the ranges of values in your sample and whether the data really do meet the assumptions of running a correlation test. All right. So I want to just cover a few common mistakes with correlation analysis before we wrap this lecture up. So one common error is trying to extrapolate the correlation beyond the range of the data. So for example, human ear length. Your ears get longer as you age. And this is real data on the length of ears of people of a whole range of ages up to people in their 80s and 90s. And you can see how like the older the person is, the longer their ears are. But does that mean we can run a correlation analysis here and get a statistically significant correlation? But does that mean that a newborn baby has ears that are 5 centimeters long? That's like the better part of a newborn baby's head. Can we extend that linear relationship to ages that aren't represented in the sample? Well, the answer is no. And you'd be surprised at how often this misconception is made, even in the research literature. So here's an example from a study from 2008. This is a study of the incidence of obesity in America, and it's been increasing since for decades now. And so this study in this figure, they had data on the kind of percentage of Americans that could be diagnosed as obese, or overweight or obese. And they had that for a few different years from kind of 1975, looks like 1990-ish, and then like 2008-ish. And in the graph, they had their observed years, and then they extended it to a bunch of projected years. Because remember, the study was published 13 years ago. So they projected from the relationship in the observed years, they kind of projected this steep continued increase in obesity up until the year, like, I don't know, 2040 or something. And they concluded that like by the year 2040, everyone in the US was going to be obese. But does that make any sense? Well, it's a big pitfall to try to extrapolate beyond your data. Extrapolation is when we try to predict the value of a response variable outside of the range of X values in the data. And there's no guarantee that a linear relationship, if it's linear within the range of X values you look at, there's nothing to guarantee it's going to continue to be linear outside of that range of X. So we know that babies don't have giant ears. Clearly, if we had more ages, we'd see that in those younger ages, the linearity of that relationship is going to change. And just because obesity is rising with a linear trend now or in the early part of the 2000s, it doesn't mean that that trend is going to keep being linear forever. We can't go beyond 100% obese. So clearly it doesn't make sense to just continue that line as a straight line. Okay, here's another common mistake with correlation analysis, and it's very much related to the idea of extrapolation. Okay, so in this scenario, we know here that Y is positively correlated with X, and let's pretend that this is our population, all of these 200 individuals here. And let's say that our sample only captured a narrower range of X values. Like you could imagine X was age or something, and we only sampled people of a certain range of ages within the broader range in the population. And if we zoom in on just that smaller range of X, we don't find a correlation between Y and X in this sample. There's no statistically significant correlation within that range of X values. We could run the analysis for these data points and we'd get an R value of 0.08, P value of 0.54 with a sample of 64. So the point here is that just because there's no correlation within the range of X values you've looked at doesn't mean that there's no correlation if you look at a broader range of X values, right? So you might fail to find a significant association over a particular range of X, even though there truly is an association if you had a greater spread of X values. So the correlation between Y and X depends on the range of X values you examine. Okay, and then one more misconception that we'll look at before we wrap up this lecture. Suppose variable A is correlated with variable B, and suppose variable B is correlated with variable C. Does that mean that A is correlated with C? Well, very often, yes. For example, people's height is correlated with people's weight. People's weight is correlated with their blood pressure, and very often height is correlated with blood pressure. But there's no guarantee that A is correlated with C. It's possible, like I've shown on this image, it's possible to have A, B, and C such that A and B are correlated, B and C are correlated, but A and C are not correlated. And so in the little example I've drawn at the bottom there, I've shown the test statistics for that particular example. There's lots of other ways that this could play out. Often, A and C could be correlated, but not necessarily. So we can't assume that. Okay, so last slide in this lecture, a practice question here. So let's say we found a statistically significant association. Well, we did, but we found this association between students' heights and the heights of their best friends. What if I converted all of the heights in this analysis to meters instead of centimeters, and then I analyzed the correlation again? How would that affect the value of r and the value of r squared? We'll take up the answer on Zoom.